Thanks, everyone. Like Alex said, my name is Abby Hogan-Brown, and I am a PhD student in the Communication Sciences and Disorders program here at Northwestern. And I'm really excited to tell you about my research, which is attempting to simplify the complex genetics of autism by studying autism in the context of a single gene disorder. So first and foremost, let me tell you a little bit about autism. Autism is a severe neurodevelopmental disorder, and it's characterized by impairments in two core domains. The first domain is social communication. This involves things like difficulty making friends, impairments in eye contact, difficulty understanding nonverbal communication, like facial expressions and gestures. The second domain is repetitive behaviors. This involves things like hand mannerisms, such as hand flapping, extreme attention to detail, and a restricted range of highly specific interests. So you can imagine deficits in these domains can have a really big impact on people's social, emotional, and educational functioning. Autism is shockingly prevalent. Current estimates suggest that 1 in 88 children born in the United States will be diagnosed with autism. Given those odds, it's safe to bet that at least a few of you here today know someone personally who has autism. Furthermore, prevalence actually seems to be on the rise. So it's becoming a top priority for both public health officials and families to identify the causes of autism and also develop effective treatments. We've known for nearly 40 years that autism is a genetically based disorder. We know this from twin studies, which have shown that identical twins who share all of their genes are more likely to both have autism than fraternal twins who only share about half of their genes. We also know that children who are born into families who already have a child with autism are much more likely to have a diagnosis of the disorder themselves. Currently, about 20% of siblings of children with autism will get a diagnosis. That's much higher than the general population risk. So while we know autism is genetically based, unfortunately, genetic researchers are having a hard time pinpointing the specific genes that are causing the disorder. We can identify a specific genetic cause in only 10 to 20% of individuals with autism. We also know that there are many, many genes involved in the disorder. A recent internet search actually showed me that there are 546 genes that have been shown to be tied in some way to autism. The problem is that only a small fraction of these genes have been found consistently and replicated across multiple studies and multiple populations. There are a couple of challenges facing genetic researchers that I want to tell you about today. First and foremost, autism is a spectrum disorder. It is very diverse. There are individuals who are really on what we would call sort of the low functioning end of a continuum of functioning. These individuals might never develop spoken language. They have severe intellectual disability, and they need constant supervision for their entire life. On the other end of the spectrum, there are individuals who are very high functioning, are only minimally impacted by autism. These individuals have a normal to above normal IQ. They're able to live independently, get married, pursue advanced degrees. This is an example of one of these individuals who's very well known. Her name is Temple Grandin, and she actually has her PhD in animal sciences from the University of Illinois. She's written several books about what it's like to live with autism, and she's spoken very elegantly about her experience. She's actually even given her own TED Talk. The problem is that many genetic studies have attempted to identify the genes causing autism by including individuals who fall all along this spectrum as one group, when in reality, it's likely that there are different genetic mechanisms coming into play at different points along this spectrum. Another challenge is that autism is a very complex disorder. I mentioned these two core domains earlier: social communication and repetitive behaviors. Very few individuals with autism have difficulties in only these domains. Many individuals with autism also have a language disability or an intellectual disability. Some individuals with autism have psychiatric issues such as anxiety or attention problems. Some individuals with autism have self-injury or aggression. And others have serious comorbid medical disabilities or medical disorders such as seizures or immune dysfunction. So any individual with autism might have any combination of these different symptoms. And what researchers are starting to believe is that different genes, which are interacting with each other and with the environment, are working in combination to cause these different symptoms. So what it boils down to is that the complexity of autism is really complicating the search for the autism genes. The goal of my research is to simplify this process, this problem, and to look for autism genes from a much more narrowly defined perspective. And this is how my dissertation is doing this. First and foremost, I'll be identifying autism subgroups, so subgroups of individuals with autism who are similar on a particular behavioral characteristic, and then comparing these individuals to individuals who have a known genetic cause for autism. In particular, my research focuses on Fragile X syndrome. Fragile X syndrome is caused by a mutation on a single gene on the X chromosome. A mutation on this gene causes deactivation of the gene and decreased production of a critical protein, the Fragile X mental retardation protein, or FMRP. This reduced production of this protein has broad, widespread cascading effects in development. It ends up resulting in atypical brain development as well as physical abnormalities. What's interesting about Fragile X syndrome in relation to autism is that that protein, FMRP, is known to interact with many of the genes that we think causes autism. We also know that approximately half of individuals with autism also meet diagnostic criteria, or who meet, approximately half of individuals with Fragile X syndrome also meet diagnostic criteria of autism. So they have enough behavioral features of autism to warrant a diagnosis of that disorder on top of their genetic disorder. So what I'll be doing is I'll be identifying these subgroups of individuals with autism and comparing them specifically to individuals with Fragile X syndrome who also have autism, because we have a really good idea of what is causing their autism. In particular, my research uses eye tracking technology to measure how people with autism and Fragile X syndrome respond to emotional faces. I'm presenting faces that are happy faces, fearful faces, and calm faces, and I'm measuring how much time my participants spend looking at different features of the faces, and also I'm measuring their pupil reaction to these faces. Now, looking time, the amount of time you spend looking at something, is a really sensitive indicator of how interesting and informative and important that area is. And your average, typically developing person is going to spend the majority of their time looking at the eye regions of these faces, because that's where we can glean the most emotional and social information from. Now, pupil dilation, or change in reaction in pupil dilation, is actually an indicator of autonomic arousal, which is the physiological response to different social and emotional information. Now, we know from a couple of recent studies that individuals with Fragile X syndrome spend less time looking at the eye regions of emotional faces. This is actually an example from a recent paper, and you can see the person with Fragile X syndrome never looks at the eye regions of these faces. We also know that individuals with Fragile X syndrome have higher pupil reactivity, so their pupils dilate more when they're looking at these faces. And we're actually thinking that this overreactivity or hyperarousal is what might be leading to some of the social anxiety that individuals with Fragile X syndrome actually experience on a day-to-day basis. We also know that in autism, individuals tend to spend a lot less time looking at the eye regions of faces. We do not yet know, however, how their pupils react to these faces, and that is one of the questions that my research will answer. So I'll be identifying these autism subgroups based on looking patterns and pupil dilation and comparing them to how the individuals with Fragile X syndrome and autism look. And then what I'll be doing is actually measuring blood levels of that critical protein, FMRP, and seeing how that correlates with these behavioral features and the autonomic arousal that we're observing in both autism and Fragile X syndrome. This research has some important implications for future studies. First and foremost, it's going to guide future genetic studies of autism. If we can identify, if we know that FMRP is implicated in autism, we can start targeting only those genes that we know interact with FMRP in studies where we're measuring behaviors that are similar to those of Fragile X syndrome. That will increase, by decreasing the pool of potential genes we're looking at, we're going to be increasing our statistical power and our ability to identify these genes statistically. It'll also inform research on drug treatments of symptoms of autism. The nice thing about Fragile X syndrome is we know so much about the genetic and molecular mechanisms that are involved that actually many promising drug therapies are currently under development. And if we believe that FMRP is also implicated in autism, there's a good chance that we may be able to use those drugs or alter them slightly to treat the symptoms of autism. And ultimately, the goal is to have more outcomes like this, where individuals are able to build upon their strengths, thrive, and pursue their dreams in ways that Temple Grandin has. So I'd like to thank you all for being here today. I'm happy to be the one to wrap up this big event. I want to thank colleagues in my lab and also Autism Speaks, which is funding this dissertation.